Okay, so welcome everyone, and thank you for virtually joining us for the latest version of the Variant Effects seminar series. My name is Mariano, and I'm a member of the BEST organizing committee, along with Johanna Becerra, C.G. Day, Mateo Cajiada, Priyanka Bayash, Yingyu Rao, and Adelaida Tovar. These seminars are made possible with support and a lot of help from Lara Muffley and Alex Hopkins, among several others, and our sponsor, the Atlas of Variant Effects Alliance. As a reminder, we are always on the lookout for potential speakers for future seminars. So if there's anyone you would like to see present, or if you are interested in sharing some of your own work, then feel free to submit a speaker nomination form. As usual, before starting the presentations, I have a few housekeeping items to mention. Each talk is around 20-25 minutes, but there should be time for a few questions. Please post those questions at any time to the Zoom QA, and we'll read them out to the speaker at the end. If you have additional questions after the end of the seminar and you are a NAVE member, feel free to visit our seminar series Slack channel for more discussion. And finally, we are posting updates on our Blue Sky account, so be sure to follow us there. You can catch the seminar today and previous presentation that have been recorded on our affiliated CMAP YouTube channel. All the details of this seminar series can be found on our websites, including a regularly updated schedule of upcoming speakers. For the last housekeeping item, this is rarely an issue, but as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. And just before starting, we would like to invite you to the MSS Symposium that is happening in May in Barcelona. So the registration is open, so feel free to register there. Today we have two amazing talks, one by Toni Beltran and the other one by Alexandra Bendel. And I'm happy to introduce Toni, that is going to be the first one. So Toni Beltran is a postdoc at Leonard Lab at the Center for Genomic Regulation in Barcelona. He's interested in understanding the language of proteins, how their sequence determines their biochemical properties and functions. In his postdoc, he has focused on multiplexing deep mutational scan of human protein domains and on combining DMS with multiple molecular readouts to understand allosteric communication in kinases. Before he joined the variant bio world, he worked on quantifying the dynamics and stability of heritable epigenetic states by sequencing many generations of isogenic worms as a PhD student at the Medical Research Council in the Laboratory of Medical Sciences of the Imperial College in London. He enjoys the combination of large-scale experimentation and computational analysis to understand fundamental biological questions. Today, Toni is going to talk about his work entitled Mutating the Human Domainome. So Toni, whenever you are ready, please start.
Thank you, Mariano, for the nice introduction. I'm gonna share my screen. Can everybody hear me well? Yeah.
Yes, that's fine.
Great. Okay. Let's go then. There we go. Okay. So as Mariano was saying in my introduction, one of the main goals of my research and in general of the work that we do in the Leonard Lab is to try to predict all the properties of proteins only from their sequence. And for a long time we've been able to know protein sequences from DNA, but the real challenge now is to really understand like how all properties of proteins and other biomolecules are encoded in their sequence. And by properties I mean any of these biophysical properties that might be of interest, like their structure, stability, dynamics, their binding affinities if they interact with other molecules, etc. Now it's also possible from a large number of sequences to basically predict their fitness. So this is because these sequences are under evolutionary constraint, and thus by sampling all of these sequences, we can get an idea of which mutations are good and they are bad. But this still doesn't tell us anything about the underlying biophysical properties that are under selection. And so in our lab, we are interested in basically this sequence to biophysical property mapping problem. And we know that all we need is data, right? And maybe DeepMind's AlphaFold structural prediction algorithms are the best example of this, where having more than 250,000 experimentally determined structures really allowed us to be able to predict structures from sequences. Now, for the rest of these properties that we would like to predict, the reality is that we don't really have this kind of massive data sets. We are only beginning to make them now. And today I want to talk about a project where we aimed at generating a large-scale data set of protein stability. But in reality, what we are measuring is the abundance of proteins in the cell. So how well proteins are expressed. And we do this using these cell-based assays where we fuse our protein of interest, which is this A here, to a fragment of a metabolic enzyme, the HFR, and then we express the other fragment that you have here in purple in a free form. And so if our protein of interest A is well expressed in the cell and is stably folded, then these two halves of this metabolic enzyme will interact and reconstitute the full enzyme, the HFR, and this allows the cells to grow in the presence of a drug. But if we introduce a mutation that destabilizes this protein of interest, then the cell typically recognizes and degrades unfolded proteins, and this will lead to basically a loss of this reconstituted complex, and in this situation, the cells are not able to grow. So basically, we are converting a protein folding problem into a cell growth problem, and so this is very scalable because we can measure these readouts for many different proteins and do it in a bulk setting. So this is how we approached this project. So we designed oligonucleotide libraries encoding many different proteins and mutations in every single position of these proteins. So deep mutational scans of many protein domains, and then we essentially transform them into yeast, and this gives us a population of yeast that express different variants of different proteins at different frequencies, and then we grow them in the presence of this drug, right? And then we can extract DNA before and after these selections, and by reading out the frequencies of its variant in its protein, then we can back calculate whether this protein was folded or not, basically generating this stability sequence to stability maps. So I guess this is for most people in this seminar. They're quite familiar with this mutational scanning approach, and this is essentially how we do it. Also, I should mention that these oligo libraries were done in a collaboration with BGI in China, because they were developing a new synthesis methodology, and we worked with them to test and to use their libraries. Right. So the goal then was to measure in a very large scale, in a very diverse set of proteins, how mutations affect their abundance. And because we wanted this to be as useful as possible for the world, then we decided to focus on proteins which are involved in human genetic diseases. And with sort of two overarching goals in mind. One, to provide this data to the community to help the development of predictive models of protein stability from sequence, but also to directly have information on whether variants or clinical variants destabilize proteins. And I think this is quite an important point to make because we have really improved a lot lately in predicting which variants are benign and pathogenic. And there's many different predictors, variant effect predictors, and they work really well. But to this day, they are not really able to tell why. So what's the mechanism of pathogenicity of a given variant? So they are agnostic to mechanism because they are basically trained on evolutionary constraint. And so we thought this would be a good starting point to say, okay, at least because we know protein abundance is relevant to the majority of, or it's a very generalizable protein phenotype, it would be cool to start measuring abundance. Right. And then the last thing I want to say before I get into the data is that because we had an oligo length restriction due to synthesis, um, we could only do up to 85 amino acid long proteins. And this is shorter than most human proteins, right? But the good thing is that human proteins are typically multi-domain, which means that they have different regions which are able to fold independently and also have their own molecular functions. And this really allowed us to cover, by splitting up proteins into these functional regions or domains, we were able to cover a lot more of the human proteome. So this is an overview of the dataset that we generated. So it's over 500,000 variants in over 500 protein domains. And they cover different structural classes. So this network representation is sort of a way to kind of lay out all of these structural types in 2D space. And so all alpha domains are in red, all beta in blue, and zinc fingers in yellow. So you can get an idea of the structural diversity that we are covering here. And then, as you can see from this in the bottom, this distribution, we have a lot of families where we only have one or two copies of a domain represented in the dataset, and then we have a few families where we have a lot of copies of domains of the same family, which will be useful to do some analysis, but I'll come back to this later. Right. So this was our experiment, so our replicate correlations after selecting these libraries, which were quite good, and then we could see pretty good separation between synonymous variants and stops, and a sort of bimodal distribution of missense variants. Similarly, we see a pretty good separation of mutation effects between mutations in the surface of these domains or in their cores. And we also saw a much stronger effect of mutations to proline, which are known to disrupt secondary structures. So all of these sort of expected trends were in the data. And then we also managed to compare our abundance scores with in vitro stabilities of many of these proteins. So we included these in the mutational scans in order to be able to benchmark the experimental approach. And we could see that we were getting reasonable correlations with in vitro data. So this is a summary of this benchmarking. So we had a median Spearman correlation of 0.73 with in vitro delta G's for 10 domains, and a median Spearman rho of 0.65 with domains that were measured previously in also in deep mutational scans, but in vitro. Right. So this is the whole data set of 522 deep mutational scans. And so what kind of analysis can we do with this? And so one of the first things that we did was to try to see if variant effect predictors are capturing these abundance changes. So in other words, how well do these variant effect predictors perform on this data? So this is the kind of correlations that we get between our abundance data in the y-axis and either fitness predictors or dedicated stability predictors. And as you can see here in the distributions, most of these performed quite reasonably. So if, for example, was the top fitness predictor on abundance, and in the case of stability predictors, thermo-MPNN, which was trained on another deep mutational scanning large-scale data set of in vitro data, was best. And then another interesting thing that we saw is that a lot of these stability predictors, which I'm showing here in blue, are actually not performing really well on zinc finger domains. And so when we evaluate them on non-zinc finger domains, the performance that we get, which is best, is again thermo-MPNN with a median row of 0.57. Cool. So another thing that we were interested in is to sort of trying to quantify where stability predictions and fitness predictions disagree. And so the idea is that these domains that are under selection for many different molecular. It's this selection is a convolution of many different molecular phenotypes. But abundance in theory should be one of the main drivers of sequence constraint. And so we thought by quantifying where these fitness predictions and abundance measurements disagree, we would be able to pick out other sites in proteins which are under evolutionary constraint for reasons other than protein stability. And so what we did was to fit these sigmoid models to this data where we compare the predicted fitness by ESM1V, one of these large language models, with our abundance data. And so if we take the residuals from these plots, we can find all these mutations which are high abundance, but they are predicted to be low fitness. And this is what they look like in a heat map fashion. So you can see that we have these vertical stripes of some particular positions with very strong residuals. And if we average them by position, we also see that they cluster around this region of this SH3 domain. And it turns out that all of these are, or a lot of this signal is concentrated around the binding interface of this SH3 domain with a peptide. So it was quite clear that we were capturing these important functional sites in these domains by doing this kind of analysis. So we did it for the entire data set. And so here I'm showing these distributions of residuals from these models. So closer to 1 means high abundance but low fitness, and at 0 it would be, sorry, whoop. At 0 it would be no additional evolutionary constraint than you would expect given the stability effects of mutations. And so here you can see that in residues or that are annotated as functional sites in these domains, these residuals are very biased towards larger values, right? So evolutionary constraint that is not explained by abundance changes. So this is if we consider all mutations. If we average the mutation effects per site, so per residue, then we see an even stronger enrichment in these annotated functional sites. And so what we did was to basically take a threshold of 0.3 here, and then say, of all of these residues which have these high residuals, how enriched are they in different types of binding sites? And this is what I'm showing here in this dot plot. And you can see that in many, many different types of functional sites, we see strong enrichments of this evolutionary signal that is not explained by protein stability. So this includes RNA, peptide, binding sites, DNA binding sites, binding pockets, calcium binding sites, chaperone interaction sites. There's a lot of diversity in this type of evolutionary constraint signal that is not explained by protein stability. And the exception is zinc binding sites, which is interesting because we know that in many zinc fingers, these zinc binding residues that coordinate the atom of zinc are important to stabilize the protein, and indeed we see a depletion of these sites. Cool, but what about pathogenicity, right? Because all over these 500 domains, we had 621 known clinical pathogenic mutations. And so the question is how many of them are explained by stability changes? And we could see that 60% of those For 60% of those, we saw a detectable loss of protein abundance. And if we set a stronger threshold of abundance loss, so at least 70%, so a decrease of 30% of abundance compared to wild type, then we had about half of these pathogenic variants. So essentially half of these pathogenic variants decrease abundance of the wild type by more than 30%. So here you can see in this density plots that a large portion of pathogenic variants are low abundance, while benign or variants that we sampled from NOMAD with an allele frequency higher than 10 to the minus 5, they are very much centered around 0, and they don't cause detectable changes in abundance. Right. So overall, half of these variants decrease protein stability, but is this equal across all types of proteins, or do we see differential enrichment? And just by aggregating across families, we could already see that in some cases, so for example, in this beta gamma crystalline domains, pathogenic variants are extremely enriched in the stabilizing mutations compared to benign variants. However, what we see is that in other families, this enrichment is actually quite small. So in these other cases, it seems that other molecular properties are responsible for pathogenicity more often than stability. And just to show you a few examples of this, in the domains where we had more clinical variants to analyze. If you look here in the left, you see these two types of domain, these two domains, the FHL1 limb domain 2. Mutations in this domain give rise to myopathies and muscle diseases. And then this TP63 sound domain, where mutations can lead to ectodermal dysplasia. And as you can see, pathogenic variants are very likely to be destabilizing, while benign or nomad variants are not. So there's a very clear separation between pathogenic and benign and nomad variants based on measuring protein stability, meaning that protein stability is the main driver of disease in these proteins. However, when we look at other types of proteins, so for example, the CRX, it's a homeodomain, so binds DNA. Similarly, SATB2 cut domain that also binds DNA, or the methyl binding domain of MECP2 that also binds DNA. We see that actually only some of the pathogenic variants destabilize these domains, but there are other pathogenic variants that don't have strong stability effects. So it seems that the mechanisms of pathogenicity are more varied in these types of domains. And so this data allowed us to classify these domains based on how much destabilization contributes to disease. And we can say that this is variable across domains and diseases. Right. And then in these cases where stability is not a strong contributor to destabilization, we could try to analyze where the pathogenic variants that are not destabilizing, where are they? And what we see basically is that they are extremely enriched in interface residues with DNA, and in second shell residues, which are residues that are directly contacting the interface residues. And we see this strong interface signal in both of these DNA binding domains. Right. And then we wanted to extend this to the entire data set. But the issue is that clinical variants are very sparse, and many of these domains didn't have a lot of data to analyze. But since large language models are good predictors of pathogenicity, then we thought perhaps we could use these predictions as a surrogate for clinical variants, and then we can extend this type of analysis. And so to try to see whether this would be reasonable, what we did is to first evaluate this on the domains where we actually have clinical data. And so here what I'm doing is comparing these Matthews correlation coefficients, which tell us the contribution of stability to pathogenicity in these domains, with the contribution of stability to evolutionary constraint in these same domains. So rather than using clinical variance, using the fitness predictions that are based on evolutionary constraint. And when we do this, we see a very reasonable correlation. And so we thought that we could use this evolutionary constraint data to try to classify all of the domains that have at least one clinical variant according to how important stability is for the fitness of these domains. And so this is what I'm showing here. So from low contribution of destabilization to disease to high contribution of destabilization to disease, we have a gradient of contributions across all of these domains. And we were interested in seeing whether this contribution of destabilization to disease was correlated with the mode of inheritance and the mechanism of diseases, at least for the domains where there were some descriptions of these. Right, and so what we were able to see is that actually in domains that contain mutations that are recessive, so they're inherited in a recessive manner, usually stability contributes much more than in domains with dominant mutations, which makes sense because these recessive mutations are usually associated with a loss of function, which is what you would expect by a loss of stability or a loss of abundance. Similarly, when we categorize this data based on the annotated mechanisms of disease, we can see that aggregation diseases or loss of function diseases tend to be better explained by stability than domains which have mutations which alter the function of the domains, and that is the reason for disease. So it was good to see that this data was capturing, to some extent, these features of these proteins. Right. How much time do I have?
Just a few minutes. I don't know how many slides, but yeah, 4 or 5 minutes more.
Okay. I will be quick with the last part. So the final question that we wanted to address with this data was how conserved are mutation effects in homologs? And this was really a good data set to do this because we had different families with at least 10. I think we had 26 families with at least 10 copies of the same type of domain. And so you could imagine that if you make a mutation in the same position, the same mutation in all these homologous positions in these domains, you could perhaps have the same effects in all of them. But you have to bear in mind that the sequences of these are actually can be quite divergent. And so there can be genetic interactions between the different residues in these domains and the mutation that you're making. And it's possible that in some cases, a mutation might have a different effect in each of these domains. But this hasn't really been quantified across different types of proteins systematically. So we thought this data was a good opportunity to do this. And when, for instance, we averaged the fitness effects of mutations by position in these proteins, and then we align them, we could see, so here what I'm showing is the mean mutation effects by residue in aligned homeodomains or SH3 domains. And you can see that there are some vertical columns here in this heat map, meaning that these mutations tend to be equally detrimental across all of these domains. But you also see horizontal stripes. So some domains that overall have more sensitivity to mutation than others. And so we were interested in modeling this data all together. And we did this using thermodynamic models. So essentially what we do is we consider this two-state equilibrium between an unfolded and a folded state. And we model this equilibrium with the Boltzmann distribution. And then for each protein family, we fit two types of parameters. So a starting stability or delta G for each individual wild type in the dataset, and then a delta G or a mutation effect for each mutation in the alignment. And this is shared between all of the homologs in the dataset. Right. So for instance, if we have these two domains that are in different starting stabilities, we could see, we do a small effect mutation, and maybe in this one we do see an effect, but because it's in the linear range. In this one we don't, because it's in the plateau. And if we make a stronger mutation, we might see effects that are very strong in both. But actually, using this nonlinear trend was important because we could see that using linear models, the data was the residuals of the models were very biased. Anyway, so this is what these models or these fits look like for example, for homeodomains as a case study. And so we could see that this model fits the data reasonably well, and we get pretty good prediction on left out variants. So this is doing tenfold cross validation, where in each of the folds we leave out 10% of the data, and then we evaluate on that left out data. So we were getting pretty good performance of 0.8 across this protein family. And interestingly, also this performance scaled with the genetic distance of these individual domains to the training data, right? So this is, rather than evaluating on the entire family, here we are evaluating in each individual domain separately. And you can see that this performance is best in these domains that have more or less 50% identity to the training set on average, whereas these that have higher divergence, the performance starts to decay. Right. And I should say this was training on this was leaving out each of those domains and then training on the rest. So the performance on these domains is on unseen data. Right. So if we then take these fitted parameters of delta G's that we get out of these models and we put them on a heat map, we could see that they match pretty well the known structural elements in homeodomains. So these prolines, for example, in the helices, and then with very strong effects in the core, but not in the surface, and so on. So we realized that this was a reasonable modeling approach, and we basically extended it to the rest of families that had multiple domains in the data set. And this gave us all these different thermodynamic models for these different families that, if we look at all together, we were getting a median performance or Pearson's R between observed and predicted fitness of 0.8. And if we take into account the experimental error, this gave us a percentage explainable variance of around 80%. Right. Similarly, when we look at the performance of these models on left out homologs, again, we were getting very reasonable predictions. So of around 0.75 or so. I can finish quickly. Yeah.
We are running out of time.
Yeah,
If you
sorry.
can, please.
So essentially, these models were working well, and so we have these average proteins describing each of these families. And so we use this to basically predict other homologs across the proteome. And when we do this again, you can see in red here that pathogenic variants tend to be more destabilizing than benign and certain variants or nomad variants, as we saw in our measured stabilities. Right. So if you didn't listen to anything up until now and you have to focus on these messages, 60% of pathogenic variants are destabilizing, with almost 50% decreasing abundance, but more than 30%. Of course, the caveat is that we are working on isolated domains, and this is a yeast experiment. The contribution of the stabilization to human disease is variable across individual domains and families, with a stronger association in recessive disorders. It's possible to combine stability data with fitness predictions from large language models to categorize pathogenic variants according to mechanisms. And then finally, I have shown you that site-wise or first-order models can explain up to more or less 80% of the variance in mutation effects on stability in protein families, even if these contain very highly divergent homologs, which basically suggests that the genetics of proteins or the genetics of protein stability is very additive. That's it. I want to thank everyone in the Learner Lab, both at the CRG at Sanger, especially Ben, and also Alex, because we've been working together a lot to continue this. And thank you for